is unable to join us for COVID reasons. But I'm pleased to be here, and I am a researcher, so I've probably got a different perspective from many of you, but I'm pleased to be here. So this session 2 is about screening and early detection of bladder cancer, and we're obviously thinking about molecular profiling and risk stratification to achieve optimal patient outcomes whilst avoiding overtreatment. So we really want to be able to identify those patients who are at high risk and need urgent treatment. So first of all, we have Peter, and I'm not sure how to say the surname, but Peter Sassiani from King's College London. So he's the academic director of the King's Clinical Trials Unit and professor of Cancer Prevention Group. And I think he's going to be talking about the grant that's been awarded from Yorkshire Cancer Research, which is led by Jim Catto, which is a 3-year investigation of early bladder screening. I'm sorry about your surname.
Oh, don't worry. Sassini, but there we go. Thank you. First of all, I think if I take a couple of minutes to try and address the last question. So I think there's probably, we need to think of a difference between a situation where you don't really suspect somebody has cancer, in which case you're trying to rule in a proportion, and what you would accept as a good enough test might be different than in a situation where you're trying to rule out the cancer and the sort of the errors. Nevertheless, in various other areas, simpler tests were introduced to prioritize endoscopy, whether that was upper GI endoscopy or lower GI endoscopy in the pandemic, and in both cases, I think people are thinking about using them far more than they did. So the cytosponge was used in people with gastroesophageal reflux symptoms, and to prioritize who should get upper GI endoscopy, which really was very, very limited at the height of the pandemic because of huge concerns about that and the downtime in an endoscopy suite after its use. And then FIT testing was used also to prioritize colonoscopies. So thank you for inviting me, Mika and Rick. Yeah, this is the trial, which is really a sort of a pilot study which Yorkshire Cancer Research have funded. Oops. So the background, for those of you who don't know about Yorkshire Cancer Research, they insistent that any research they fund has to have the potential to benefit the people of Yorkshire in a reasonably short time period. So one of the reasons for doing this in Yorkshire is that Yorkshire has the highest incidence of bladder cancer in England. The disease-specific survival, as you all know, is not really improving, although that does depend a little bit on how you define bladder cancer and how much you include. But the mortality is not improving. Most bladder cancer is diagnosed in people with visible hematuria or non-visible hematuria. The potential to use hematuria to detect asymptomatic disease seems to be there. I'll say, yeah, there have been a number of studies that suggest that there might be something there. It's just no one's ever been able to put together something that makes sense as a screening program. So previously people have said that it looks like you could reduce mortality from bladder cancer if you used hematuria in a screening program to be cost effective, or to be also to get the benefits to outweigh the harms of over investigating. You're going to have to target a high risk group. So the dipstick hematuria, it's cheap, it's simple, it can be done at home, and it has the potential as a screening test. So we're now using an app so that we can actually read the tests using a mobile phone and a camera, but although that it's not approved, the regulatory issues means that we're not really using that. So I'll talk about that in a little bit. But the problem is, as I think it's been said earlier today, that having non-visible hematuria is not, the risk of cancer with non-visible hematuria is less than the 3% criteria for referral under a 2-week wait guideline. So how do you use it? Well, how are you going to triage the non-visible hematuria to select out a group who will really benefit from a diagnostic investigation? And cytoscopy is too expensive and too invasive to use for everyone who has non-visible hematuria. So urine cytology and ultrasound are cheaper and less invasive. And so one of the things we're going to do in the study is to kind of reverse the process. And so we will use those as triage to see who should get cystoscopy, given that these are all asymptomatic patients or people. And then we hope that's going to be a much more efficient, and would mean that it could be a reasonable screening procedure. Undoubtedly, there will be people who have cancer that is missed, but at the moment, everyone's cancer is missed, if you like, at the point that we're doing this. So it's can you, for a substantial proportion of the population, advance when their cancer is diagnosed and hopefully find it before it is muscle invasive. And examples, both the cervical screening cytology that was used until recently in most of the world probably only had, it probably only had a sensitivity of about 50%. And the guaiac bowel screening test probably also had a similar sensitivity, or even slightly lower. And yet both of them made a big difference if you put them into a properly organized screening program. And then we'll be focusing on individuals who we think are at high risk in some sense, and that's either because they've got high risk of lung cancer and they've been identified through the targeted lung health check or the Yorkshire Lung Screening Trial, or that they are living in areas with high rates of bladder cancer mortality. So based on geography, we can identify people as being at high risk. And 2 previous studies, both had reasonably promising results. Neither of them are what you'd call high-level evidence that you could use dipstick hematuria for screening. But one of them found 17 bladder cancers in just under 2,500 men, and those bladder cancers had reasonably good prognosis compared to the normal. And in the other 1,500 men in America, and none of the men who had bladder cancer diagnosed through the screening died from bladder cancer. And in a comparable group, they would have thought that about 15% of them would have died. So some reasonably encouraging, but not very strong evidence. So we want to sort of see what is the feasibility of doing a large trial of bladder screening. And so we're going to look at the home urine dipstick test in the first place, and then triage that with cytology and ultrasound. We're going to look in 3 different populations in order to decide whether a large trial is feasible what population would make most sense to do that trial in, and can we use cytology and ultrasound to triage who gets cystoscopy. So everyone who takes part will have a urine self-sampling to examine the presence of hematuria and glycosuria. I will explain the glycosuria in a little bit. And in 2 of the cohorts, those with hematuria or glycosuria will be sent to an early detection clinic for further evaluation. So that's with the ultrasound and cystoscopy if they've got hematuria. And then we'll be following everyone up in the National Cancer Registry. So the dipsticks, there are 6 dipsticks in a pack, and we hope that people will use at least 3 of them. If you're positive on any one, they'll be referred. So the first cohort is in people who are already in the lung screening trial, and then the second is just men rather than men and women, and in geographies where they have high rates. So the primary objectives are to look at the acceptability of urine self-testing and early detection clinics, compliance with the self-testing, so how many of the dipsticks will actually be used, whether it's okay, what is the prevalence of hematuria and glycosuria in these people, are they happy going to early detection clinics, and what happens in those who are referred to those early detection clinics. We're going to be looking at symptoms within the population, so that they'll be asked about symptoms when they do their dipstick tests. How much bladder cancer do we actually find? How accurate are cytology and ultrasound compared to cystoscopy? And recruitment rates, so how big would a big trial really have to be if we're going to recruit enough people? So this is these tests. So they've got 5 channels. It's the Roche-Comber test strip and supplied by test card. So we're sending 6 strips in a little pack. And there's a mobile app which is adapted from test card's app, but made specially for the trial. So you can take a photo of the strip with a camera, and that would read the signal. But because it's not approved yet, we're actually having to get a human to look at the photo rather than use the AI algorithm. But we are blinding people to the different channels, so unless they go and look it up on the internet, and we're only telling them about one of these strips. I'll come to that. So everyone gets a questionnaire, symptom questionnaire, and then they're told that they're going to be randomized. So they're randomized to either having the hematuria or the glycosuria being told to them. So we will, if they have positive hematuria and they're in the hematuria arm, then they go on to get referral. And if they've got positive glycosuria in the glycosuria arm, they go on to get referral. So in the cohort 1, which is the Yorkshire lung screening trial, everyone in this study has already been randomized to the lung screening arm. So we have a control group, if you like, but they will get no screening. There are controls already for the Yorkshire lung screening trial, and we can find out about bladder cancer incidence and mortality in that control arm because there are already controls for this other trial. So we're just doing hematuria in this group, not looking at the glycosuria, and we want to see So these are people who probably will take part because they've already agreed to take part in lung screening trial. When they come for their second CT scan, they will be given information, given the test kits, but they'll have to take it home because there isn't facilities for using toilets on the mobile units. There aren't many toilets, so they don't want everyone to try and be using a dipsticks on those mobile units. And we want to recruit 800 people. It'll be both men and women, but they all will have had to be eligible for the CT scan based on mostly smoking history and age. And then if they have hematuria on any of the dipsticks that they use, they'll be referred through a 2-week wait pathway. So it's the same randomization that's already been done. So that's why everyone's going to get hematuria testing here. In cohort 2, where we're going to send 3,000 people kits at home based just on geography, and so that will be just in men because of the higher risk of bladder cancer, even though survival is worse in women. We've decided that this should probably just be restricted to men. And they will be, as I say, randomized to either looking at the hematuria signal or the glucosuria signal. And if they test positive to whichever one that we're looking at, they will be referred to a special clinic. The GPs were not keen to have people with glycosuria sent to them. So we've decided we'll keep that in our bladder detection clinic, and we'll also be doing that. So if you've got glycosuria, you'll get a blood test for HB1AC, which is sort of a standard test for type 2 diabetes. And I think it's reasonably likely that we will find a fair amount of type 2 diabetes that is undiagnosed. And so we can also look at the benefit, but it's a reciprocal design, as I'll come to. So if we had just randomized people to dipstick or no dipstick, I think the uptake of the dipstick would have been pretty low, and that reduces the power considerably because we've got to look at everyone randomized to being offered a dipstick to be everyone randomized not to get one. If only 40% of the people who we send a dipstick to actually return the dipsticks, having used them, or return the results, we would have very little power to distinguish what was going on. So we wanted everyone to actually sort of say, yes, we're happy to use a dipstick. So we're only randomizing those that do. It's really difficult to get a placebo dipstick. Nobody makes one. It would have been extremely complicated. We didn't think it's really fair to say, okay, we're just doing a dipstick and we're totally ignoring the result. We're just looking for hematuria, but we're not going to tell you what's going on. I don't think many people would have liked that. So we felt, well, actually, if there's a reciprocal, we're telling you, you're either going to be screened for bladder cancer or possibly for diabetes, and you'll be a control in one study and you'll get the screen for the other one. That would be more acceptable to patients, to participants. So that's what we've gone for, this reciprocal design. We've had a whole discussion, in fact, with Phil, about whether or not to return the results at 12 months for the other test. Actually, there are 5 channels. We've got no intention of telling people about the other 3 at all. And we, after a long session with clinicians and patients, it was agreed that it doesn't really make any sense to tell anyone whether they had hematuria or glycosuria 12 months ago. If they've got symptoms, they should be talking to the GP and getting it tested because of symptoms. If they haven't, all anyone would do is presumably repeat the test. And that starts to dilute the effect of screening if in the control arm everyone gets screened, but a year later. So that's not going to happen. But big ethical questions about, is it okay to ignore a test result? We had hoped that this would be all done by the app, so no human would know the test result. It would just be in a computer, and it'd be like having a blood sample that you haven't tested, you know, or an image that you haven't yet looked at. But in fact, you know, we will have to talk through and see what's going on. But because neither of these, in the absence of symptoms, are actionable tests, you know, ethics has agreed that it is ethical to do this, and it's, you know, it's a little bit like giving a patient a placebo, because you don't know there is any benefit of giving them that treatment. So there may be no benefit of telling people that they've got hematuria in their blood. So, yeah, in other studies, we would store the sample and do the testing at the end. Unfortunately, we can't do that, but there are, you know, there are other power advantages by knowing we can go back and look at exactly who had hematuria in the glycosuria arm, and we'll be able to say in a much smaller group, did this make a difference? Because these patients had hematuria and we acted upon it, and these patients had non-visible hematuria and it was ignored. And so you could get a much smaller study to answer the question about, does intervening by referring to cytology and ultrasound reduce the chance that they get advanced muscle invasive bladder cancer or die of bladder cancer? So I think I've run over time, but the third cohort is just to look at the sort of sensitivity and specificity of the different tests. So this will be in people with 2-week wait referrals, and in the 2-week wait clinic, we will be doing a dipstick. So there will just be one dipstick probably, but we'll try and get more by doing it whilst they're waiting for referral, and comparing the ultrasound and cytology to cystoscopy. Thank you.
At the end, so can save any questions for then. So next, I'm pleased to invite up Mika and Rick, who are going to talk about BC Recon, which to me was something about reconstruction, but apparently not. So.
Thank you. So this is a project that we're very pleased about because Rick and I got this funded by Cancer Research UK. So this is a work that will start hopefully on the 1st of June. So we got about £650,000 from Cancer Research UK. And obviously we're always all pleased when we get a grant, but the reason we are so pleased is because we know it's so hard to get money for bladder cancer because it's not one of the prioritized cancers by Cancer Research UK, despite the fact that it's amongst the 10 most common cancers. So we think this is a really big step forward, and we're hoping that this will also help us when applying later in the year to the MRC to get funding to help BC Track, which is a consortium Lydia talked about earlier this morning. So BC Reckon, it's a much shorter term for this very long title. So diagnosing bladder cancer, evaluating the role of a urinary biomarker test in the reconfiguration of hematuria clinic investigations. So this follows nicely from everything we've already heard this morning when Ramesh was also talking about, are there other things we can do to help with diagnosis of bladder cancer in COVID times? So we already talked about one urinary marker. So Rick and the team in Birmingham, and we'll come to that in a second, developed another urinary biomarker. And so we're going to do a study now to kind of see how that may help in future diagnostic settings for bladder cancer. And I think this slide is potentially already a little bit unnecessary because we have talked about it a lot already. But I think it's interesting to note here that really only 1 in 5 adults with visible hematuria and about 1 in 20 with non-visible hematuria ultimately present with bladder cancer. So the point that we're trying to make in this slide is actually there is a lot of investigation that has to happen to then find the people who have bladder cancer. And so it's trying to kind of improve the experience for the patient, irrespective of whether they have cancer or not. And obviously, if we can come up with a non-invasive alternative to cystoscopies, the urinary biomarker would be very welcome, because at the moment, so this is what the NICE guidelines say, a GP will refer any patient that fulfills these criteria to a hematuria clinic. So it just shows we're not very precise yet. We can't really specify who really has to go and have all these investigations. So there's a lot that needs to be done still to make the experience better for the patients. And so when it comes to the diagnosis of bladder cancer, obviously we've talked about the cystoscopy, which is the invasive way. You have the dipstick, well, Peter was referring to as well, but you could have an ultrasound CT scan. We could look at the cytology. And ultimately, I think an important point here is that 80% of all the patients will be discharged with no normality. So we do need better ways of diagnosing bladder cancer, and preferably also earlier. And so this is where Rick will now tell you a bit more about the urinary biomarker, as well as the setup that we have in this specific study.
Yeah, thank you for that great introduction, Mika. So yeah, I'll just come back to this. 80% of patients who attend a 2-week wait hematuria clinic will have no abnormality detected, and they will be discharged back to primary care. And we're investigating up to 200,000 hematuria clinic patients per year in the UK. And so 160,000 patients will undergo imaging urine cytology. But worst of all, as I'm sure patients here will report, is the flexible cystoscopy, which is invasive, and it's also very expensive for the NHS to provide. It has to be undertaken by, in most cases, it's undertaken by a clinician. There's a huge amount of behind-the-scenes sterilization equipment needed. The kit itself, a flexible cystoscope itself, costs about £12,000. The equipment stack to run it costs about £100,000. So it's burdensome for patients, and it's very expensive for the NHS to provide. So for years, as we've heard earlier from Ramesh, and who covered one of the systematic reviews of urinary biomarker tests, for years, there has been an endeavor to identify, develop accurate, sensitive, and specific urine tests for diagnosing bladder cancer that could avert the need for some cystoscopies. And obviously, a urine test that's as accurate as Flexi means that fewer Flexis would need to be carried out in hematuria clinic. Clearly, that's beneficial for patients and the NHS. So reduces costs, reduces patient burden, and potentially means that waiting times can be improved. And so with a previous grant from Cancer Research UK, a biomarker project award, we worked for several years on the development of a urine test that could replace some flexible cystoscopies. And working with Doug, who's at the back of the room, who asked the difficult question to Ramesh earlier. So working as a team, we have devised, developed, validated, and commercialized a new urine test for bladder cancer. Okay, and we now want to test our test prospectively in patients undergoing investigation for hematuria. It's an observational study, so we're not changing the pathway in a similar way to Peter's study. We're observing what happens to these patients with the addition of a urine test alongside imaging, urine cytology, and flexible cystoscopy. And that is what the BC Recon early detection and diagnosis funding is for. Can our urine test reduce the number of normal patients who undergo flexi and so reconfigure hematuria clinic investigations? The premise being that if a patient is negative on imaging, is negative on urine cytology, and is negative on our urine test, so triple negative, to coin a phrase from another cancer setting, if a patient is triple negative, do they need a flexible cystoscopy as well? So this is the sort of overview of our urine test. So our urine test relies on the DNA that's extracted from a 30 milliliter sample of urine. Okay, we need 25 nanograms of DNA, and we're looking for mutations at 443 different parts of the DNA in 23 genes. And those genes include the TERT promoter, FGFR3, and KRAS that the Euromonitor test includes, but also 20 more. And any single mutation at one of those 443 points is a positive test. Okay, and with that approach, with a level of thresholding to remove background noise, we achieve 87% sensitivity and 85% specificity for the diagnosis of new cases of bladder cancer in patients undergoing investigation for hematuria. And that sensitivity is compared with a final diagnosis following transurethral resection. Okay, and so those figures would indicate that the urine test is as good as flexible cystoscopy. And you can find the data underpinning these figures in poster 3 and also shortly in European Urology Oncology. Now, the other thing that I was sort of chipping in earlier, which Rob Jones entirely appropriately stopped me from running away with, is that there are a couple of interesting aspects to the test. So we've designed it to diagnose disease. That was our premise to start with. However, FGFR3 mutations, we know that patients who have those in their tumors and hence in their urine DNA, those patients tend to have low-risk non-muscle invasive disease. TP53 mutations, which are also in our panel, would at least double your risk of being diagnosed with muscle invasive bladder cancer or high-risk disease. And so there's already an opportunity to potentially risk stratify patients for the rapidity of their urgent investigations. So some might be investigated more urgently, some less urgently. That's not what we're investigating in the study, but it's something to think about in the future. Other mutations in the test might also be very useful. ERCC2 mutations. Data would suggest that those mutations predict complete response to platinum-based neoadjuvant chemotherapy. So that's interesting. So, as I said, we'll run our urine test in addition to the standard care in hematuria clinics. We're going to recruit at least 3,200 hematuria clinic patients over a 3-year period, and hopefully to obtain around 400 bladder cancer patients. And we will test the urine of those 400 bladder cancer patients, as well as a random selection of 400 patients without bladder cancer. We'll also, now clearly with we do get some false positives represented by the specificity of the test, and we do get some false positives, false negatives represented by the sensitivity of the test. So we will be following up all of these patients for at least 1 year subsequently, possibly through NCRAS in a similar approach to Peter, to find out if they are subsequently diagnosed. So patients who are negative, actually, were they subsequently diagnosed with bladder cancer and hence were false negatives? Or if they were positive, but with no cancer on the day, were they subsequently diagnosed later and so actually weren't false positives, were probably true positives? Okay. And clearly, you know, as I outlined at the beginning, our test will hopefully have a significant impact in reducing the number of patients investigated in hematuria clinics who come away with no diagnosis. If we can at least halve those patients with no abnormality detected who undergo flexible cystoscopy, that will be a big achievement, both on the burden for patients, cost to the NHS, and waiting times. You know, and that's particularly pertinent, prescient at the moment, given the waiting times that we know exist as a result of the COVID pandemic. One other thing to say, clearly another great priority for bladder cancer patients, non-muscle invasive bladder cancer patients, are those who are undergoing surveillance for their disease following their initial diagnosis, treatment, and in most cases, intravesical therapy. So can our urine test help to reduce the number of cystoscopies that those patients undergo? Well, yes and no. It remains work in progress. So for patients undergoing surveillance, we get the same level of sensitivity, 87%. So we're detecting 87% of all recurrences, but we have a much lower specificity of around 65%. So we have a lot more false positives. Okay, so our test is positive, but without there being disease seen by cystoscopy. However, when we look at those false positive patients, and again, follow them up for up to 18 months, a significant proportion of them are subsequently diagnosed with bladder cancer, such that if you're a false positive test, you're 3 times more likely to recur in the future. Okay, so we're almost preemptively diagnosing bladder cancer, or where the urine test is diagnosing some cancers before they're visible by cystoscopy. Thank you.
So thanks, Rick and Mika. Very quick question. So you talked, both the last speakers have talked about hematuria clinics, but are there patients with bladder cancer, diagnosed with bladder cancer, who don't have hematuria, either visible or non-visible? And hence, is this the right population? Okay, so next we have a change in the program, and we have Vicky Sales, who's representing Fight Bladder Cancer and is standing in for Melanie Costin. So thank you.
Hi everyone, I'm standing in for Melanie today, as just been mentioned, as she's not very well at the minute. But my name is Vicky Sales and I work for Fight Bladder Cancer in the communications team. Today I'll be delivering a short presentation on the patient and care experience of non-invasive muscle bladder cancer surveillance. Fight Bladder Cancer has a vision that everyone will survive bladder cancer and live long and well. Our mission is to lead the fight against bladder cancer. Our work is driven by patient and family insights. We are patient-led, compassionate, action-orientated, and ambitious in terms of our values. Here is a picture of my dad. He's living with stage 3 non-muscle invasive bladder cancer. He was diagnosed at 44 in 2016. He has always said that the most challenging part of his cancer journey is the waiting. Perhaps we could define this as anxiety. This is the feeling that patients, like my dad, get in the time leading up to a scan. This could be when they're waiting for an appointment for their scan results, or generally at any time when thinking about a procedure turns a mainly rational brain into a mess of doubts and worries. When they get certainties that there is pain here and ache there, and that surely there is something terribly wrong somewhere in their body. It is very common for people with bladder cancer to experience a dip in mental health with feelings of depression or anxiety, but this does often reverse as treatment progresses. But there are solutions to help. This includes cognitive behavioural therapy, antidepressants and anti-anxiety medication, and prehabilitation such as physical therapy, counselling, addressing risk factors, and social support. Here is some advice my dad gave to cope with surveillance. Firstly, he said to try and keep busy. My dad started a football team, and we also got a dog called Dolly to encourage him to get outdoors and walk more. Secondly, he says to be organised with your treatment. He says it was also important to surround yourself with love, and he also encourages patients to get in touch with those in similar situations. Fight Bladder Cancer allowed him to meet and speak with people going through the same thing as he was, which helped him a lot. Here are some results from a 2019 global survey of 1,615 bladder cancer patients and their carers from 39 countries. It shows that blood in the urine was the first symptom reported by over 72% of respondents. However, 3/4 reported that their initial diagnosis was of a condition other than bladder cancer, which we see happens all too often as a charity. And shockingly, over half of the respondents had never heard of bladder cancer before their diagnosis. The survey also found that the most popular support form was friends and family, followed by online support groups. We can see the popularity of online support groups from our own, with over 5.4k members. The National Cancer Patient Experience Survey is another survey conducted yearly in England and asks patients questions about the care they have received during their cancer journey. Here are the results from the 2019 survey. From them, we can see that the treatment of bladder cancer patients in medical settings is less than satisfactory. It showed that roughly twice the proportion of bladder cancer patients compared to all other cancer patients said they were not given the name of a clinical nurse specialist who will support them through their treatment. We can also see that bladder cancer respondents were roughly 20% less likely than all other cancer patients to have been given information about support or self-help groups. As well as this, they were less likely than all other cancer patients to have discussions with staff about the impact that cancer could have on their day-to-day activities, to have someone talk to them about their worries or fears as an outpatient or day case, and less likely to receive information about financial help or benefits. Clearly, we still have a lot of work to do to improve the lives of bladder cancer patients. Thanks everyone for listening.
We wish your father all the very best. So next we have Nada from University of Aberdeen, and Nada is online, I think. Do I have to...
Hello, can you hear me okay?
Hello, Nada. So Nada is a third-year medical student who's an aspiring surgeon and a research enthusiast, and is going to tell us about implementation of bladder biomarkers in bladder cancer. Thank you, Nada.
Thank you. So good afternoon, my name is Nada Judah and I'm one of the third-year medical students from the University of Aberdeen. I'm very grateful for this opportunity and really happy to be here. I'm very sorry I cannot be there in person, but today I'd like to share with you my summer research project on the current status of biomarkers in bladder cancer. So this is what I'm hoping to cover today. In the next wee while, we'll touch on the aim of my project, have a look at the background of bladder cancer, the structure of my project, what were the relevant findings, and then finishing up with some conclusions. So let's get right into it then with the aim of the project. My goal was to systematically review what the barriers are, so what's stopping us, and the facilitators to allow for successful implementation of diagnostic biomarkers in bladder cancer management. A little bit of background to bladder cancer. So this is a major global health problem with high incidence. Just in the UK, there are over 10,000 new cases of bladder cancer. It also has a really high rate of recurrence, an astonishing 73% in patients with high-grade disease. Another important factor is its poor prognosis. It has a very high associated mortality and a low one-year survival rate, and this is particularly in patients with high-grade disease, as this is often how people present with due to its late onset. So this slide just shows my screen is frozen, unfortunately. Can you hear me, or can you hear me okay, or? Oh no, my screen is frozen. I'm very sorry about this, just technical issues.
We can hear you. I think you've kind of skipped on towards the end of your presentation. Everything went through. Yeah.
I'm really sorry about this.
Brilliant.
It's frozen again. I'm sorry. It's going to be a little bit slow.
So Nada, when you clicked through the slides before, it did move on, but just it went through very rapidly. Have you tried doing the same again?
Unfortunately, I'll try. Should I stop sharing and then reshare, because my slide is frozen?
Yeah.
I'm really sorry about this.
Maybe this one slide's the problem, so maybe restart again on the next slide. Do you need to reboot your? Yeah. Do you want to email us the slides, Nada?
Okay. I have shared my screen. Is it sharing or not yet?
Not yet.
Okay.
Yes, you are now.
Okay. I'll try it again, and if not, then I will send my slides.
Okay.
Okay. So I think I was on this slide, and this slide just shows the approach to management of bladder cancer. I'm really sorry again. I hope it won't happen again, but this slide just shows the approach to management of bladder cancer, and the key thing here is that currently cystoscopy is the gold standard for the detection, diagnosis, and surveillance of bladder cancer, and biomarker use is really restricted to clinical research. And the question is why? Why is biomarker use not recommended and not implemented into clinical practice when it's a much cheaper and less invasive alternative to cystoscopy? So most of the studies looked at urinary biomarkers, as they're very easily obtained and directly originate from the site of pathology. But there's many other types of biomarkers, for example, DNA, RNA biomarkers from the bloodstream, and more types of biomarkers, as you can see on the slide. So how did I investigate this topic? Well, a systematic search was performed from inception up to the 7th of July 2021 in these following databases. The search terms included bladder cancer, biomarker, and implementation. The articles were screened against the inclusion and the exclusion criteria, which were that they must be about bladder cancer and biomarker, and not any other type of cancer or investigation, and they must also include mention of implementation. No primary implementation studies were identified, and so this review focused on the available expert commentary articles to explore what the barriers are and the facilitators to biomarker implementation. Theory-based analysis was conducted using the implementation framework of choice called CFR, and this stands for the Consolidated Framework for Implementation Research. It has a wide variety of different constructs, and this is just a screenshot just to show you some examples of them. The relevant constructs were selected and put into an Excel spreadsheet and had their definitions slightly adjusted to match the content of the articles. And each construct was then given a particular color. And this is how my final data extraction sheet looked like. Again, a screenshot. The included articles were highlighted in green, and the ones that were discarded were highlighted in red. And the columns focused on me gathering information surrounding the intervention biomarker itself, the patients, and also the healthcare system in general. So as I read the articles, I coded them against the CFR constructs shown earlier. I then extracted the relevant data and then gave this data the color that matched that particular construct. So what were the overall findings then from the project? So systematic searches returned 240 studies, and after abstract screening, 65 remained. Full text screening resulted in a pool of 30 included articles, which were data extracted and coded against CFR. The analysis showed that the most frequently occurring construct among the articles that would need to be addressed to allow implementation was intervention characteristics, namely intervention source, evidence strength and quality, and complexity. 1A refers to the fact that the biomarker intervention is not yet fully developed. 1B is to do with evidence that is collected about biomarkers, and 1F, which is complexity, refers to the fact that the method of detecting the biomarker is complicated and not yet practical. If we have a look at the barriers in a bit more detail, taking intervention source first of all, the majority of articles concluded that the available biomarkers lack sensitivity and specificity, and that implementation of these biomarkers would only be feasible if the patient would accept the reduced sensitivity, which in most cases they wouldn't. They would still opt for the more invasive cystoscopy, as that would be more reliable in giving them their correct diagnosis. The other big construct that came up frequently is evidence strength and quality. The articles highlight that evidence for biomarker use is limited due to the insufficient sample size and the small populations used in the studies. Furthermore, although protein profiling studies have led to the discovery of promising biomarkers, majority of these studies remain to be validated in large independent sets of samples. And validation is required to determine whether these biomarkers represent a significant addition to current management of bladder cancer tumors. The third construct, which came up again quite frequently, was complexity. It was found in the commentary articles that there are technical issues regarding the profiling platforms used, specimen type and preparation, and also tissue heterogeneity. Bladder cancer was found to be highly heterogeneous, i.e. extremely varied in presentation and structure between individuals, and this would cause difficulties in detecting single biomarkers, so a panel of biomarkers may be more useful. And these here are the biggest areas found to be facilitators for biomarker implementation, and they could have some potential for the future. One of the areas that was highlighted by the articles is the establishment of a multidisciplinary panel of experts, guiding the proteomics investigators along the multiple turns and bumps that follow biomarker discovery. Joining clinical experts and biomarker investigators can play a catalytic role in accelerating the biomarker implementation pipeline. Moreover, agencies need standardized guidelines to ensure that molecular marker studies conform to basic standards in design and reporting. And the beginnings of this is very much underway with the development of reporting recommendations. It is also necessary to have improved definitions of the disease communicated on a much wider basis. This is to avoid false positive results and leads to more effective use of these tests for cancer detection. The establishment of a collaborative network would clearly facilitate these essential interactions. It would also improve data sharing among the scientific community. It is important to tackle biomarker validation and implementation in a more spherical manner, as there exists fragmentation and isolation of research efforts, and biomarker validation and implementation cannot be efficiently addressed in solitude. Having a network-driven approach with a central funding mechanism could really address some of the challenges posed and pave the way for future implementation. The findings of the study have to be seen, however, in light of some limitations. The biggest limitation of this study is that there were no primary implementation studies, which means that the data we have collected is really a collection of expert opinions and hypotheses on why biomarkers are not yet implemented. It's not actual data or evidence directly derived from studies. We could argue that this field hasn't developed yet to warrant those implementation studies. And this is in fact really important to know, as we have identified some gaps in the literature and gave possible explanations from expert opinion leaders as to the reasons why. The second limitation concerns commentary articles. Since they are opinion articles, there could possibly be some bias in them. Authors might have interest in pursuing biomarkers and thus have written very favorably about them. And the opposite would also be true, for example, if you have any conflicts of interest. So what's the overall take-home message then? Well, it was found that the major barrier to biomarker implementation still lies at the intervention level, and more work is needed at the translational research stage before successful implementation can take place. Thank you very much for listening. That's the end of my talk. I'm very happy to answer any questions.
Okay, so we now open the discussion to the audience. I think we've got some feedback. Is that the case? No. Okay. So open to discussion on next steps for non-invasive diagnosis. So Rick.
Hi there. Question for Peter, actually. So somebody probably ought to run and get Peter's senior microphone.
I will just remark that when I first moved to Leeds in 1990, the urologists had just recently performed a hematuria screen of many people, and they were so overwhelmed with the number of people they'd found that they quickly dropped it. So, yes.
Just about the dipstick testing, you're asking for minimum of 3 tests undertaken out of 6. Are you specifying a time period or separation for those sequential tests?
So we're asking them to do daily for 6 days, and then we'll be, you know, if they do all 6, that's great, but we're hoping that we'll get sort of 3.
Yeah, I mean, I think there's potentially some interesting data on the continuing hematuria, if you like, the persistent persistent non-visible hematuria.
Yeah, I mean, it will be interesting to see, you know, of those people who do all 6, you know, the consistency of those results, and whether it's more important if they have all 6 positive than if they just have 1 or 2 out of 6 positive.
Yeah.
So we'll hopefully get that, but you know, we don't expect 90% of the people who do 1 dipstick to do all 6. So it's sort of known from bowel screening that when you needed 3, it was really unpopular.
Okay.
And the proportion of people who return a single FIT is much greater than the proportion who returned the 3 guaiac.
Thank you.
Peter, could I ask, while you've still got the microphone, so you mentioned that some of the patients will be screened for type 2 diabetes, and I just wondered what happens, because presumably in Yorkshire there's quite a high proportion of patients who will already have type 2 diabetes diagnosed.
So they're eligible. So they won't. So one of the things is, we will actually see whether the people who it's known, are we picking it up? It's not considered to be a screen for type 2 diabetes, and the endocrinologists would get very cross if we called it a screen for that. So it's sort of a screen to see who you will do a screen in. People are aware. We wanted to call it a urinary health check. The whole question of how you propose the thing, but we think that there could actually be a fair amount of undiagnosed diabetes in Yorkshire, and that this would be potentially a way of finding some of it. So the primary interest, and certainly if the funder is in bladder cancer, but I think if it'll be interesting to see what happens, and because the blood test is definitive, I don't think there's any worry about overdiagnosis of diabetes. That being able to intervene early should improve the health and the quality of life of those people if they're really found to have diabetes. But
presumably it's not going to really address the underlying question of why there is a higher rate of bladder cancer and more aggressive cancer in the Yorkshire population.
No, we're not doing an epidemiological study, and certainly not a comparative epidemiological study. I don't know that there is a higher, I mean, so the survival being worse would be linked to a later stage of diagnosis. I think probably a lot of it is industrial, but there'll also be high rates of smoking. So we're not looking for new risk factors, and the occupational information we get won't be amazingly good. So we're not going to be able to sort of assess exposure to different carcinogens through work. We'll just look at sort of large categories of occupation.
Mika.
I have a question for Nada. So you went through the literature, Nada, obviously specifically focused on bladder cancers, but are there any other settings where in another cancer or another disease where we can learn some lessons from when it comes to implementing biomarkers instead of perhaps using anything that gives us more visual evidence of a cancer being there?
Definitely, there were so many other cancers in the literature, urology cancers, for example, penile cancers. This could be quite helpful. And it was also in the female population as well, endometrial cancers. There's lots of biomarkers out there that we could potentially use. And it's something that's really non-invasive and cheap and could really help diagnose things easier for people.
Sorry, I'll rephrase my question. So I'm aware that we can use what we find in, what we do in bladder in other settings, but I'm wondering, is there already a setting that we in bladder can learn from where they're ahead of us in terms of implementing biomarkers?
Yes, I understand your question. There is a setting in prostate cancer, but they haven't yet implemented biomarkers yet, as far as I'm aware. So there's lots of biomarkers out there for prostate cancer, but it's not yet in that same stage of implementation.
Rob.
Yeah, so this is probably another question for Peter, but I'm interested in anybody's views on it, because I guess if we're setting out on a screening program, are we going about it the right way by by focusing on a given biomarker in a given disease, because presumably ultimately screening programs will be multiplexed. And you're not going to go separately for your bowel screening, separately for your bladder screening, separately for your prostate or breast screening, separately for your endometrial screening. Do we need to be looking at the end game in a bit more detail now in order to get the technology and the collaborations correct for successful delivery, not of a screening program that has all the right specificity and sensitivity specifically for bladder cancer, but one that will fit into a practical and workable socioeconomic solution?
Can you answer that?
Peter's looking the other way.
So as you know, that I'm also running the NHS Gallery trial, which is looking at a multi-cancer early detection test in a screening setting, which essentially looks for cancer, full stop. They've shown that they can detect 50 different cancers. The first step is essentially a pattern which is common to many different cancers. So it's not site specific. I think there's a lot of interest on what you would then do. So that test is very specific. So only 0.5% of people without cancer test positive on it, but lacks sensitivity. So you might want something that's more sensitive for a particular cancer. But in any case, we would probably want to think about what appropriate triage tests. If, as we think, that test has a positive predictive value for bladder cancer of 50%, then I guess you'd be very happy to do cystoscopy in all those people. But I think it may come too late. And so we need to think, well, there might be a future multi-cancer early detection test which is rather more sensitive to early cancers, but doesn't have the same specificity. And then to think about how you'd use biomarkers to triage. But I wouldn't want to sort of stop research into early detection of individual cancers because of the promise of a pan-cancer test, even though I think you're right, we need to think about how those would all integrate.
So are you saying that it's, so ultimately, I guess, let's just say the Gallery test is the thing, that actually should we be now trying to look at developing the triage test for, so in other words, your starter population isn't male smokers or whatever, it's actually people with a positive Gallery test. Should we be working on that now? Or are you saying it's premature?
Well, so I say, if the Gallery test is the test, then we probably don't need to triage Gallery positive. You could just go straight to a flexible cystoscopy, because if the Gallery test is the right thing, the positive predictive value of a positive Gallery test will be very high. If it's something that uses a similar technology but is not the Gallery test, then it may well be that we need a triage test, because you would be, the worry with the Gallery test is that its sensitivity for early cancers is not great. And so will it give you enough of a lead time, and will we want to be doing more? So I think thinking about how you would combine all those things, people want to think about an overall specificity, and say, oh, that's not acceptable that if 10% of people are positive, because you're doing 5 different screening tests, and it could be much more, it might be 30% of people would have a positive screen at some stage over 10 years if you're doing lots of different screening tests. But I think it really depends on what happens next. If the next test is just a blood test or a urinary test, then it doesn't matter, particularly if you could combine the two. So, but or you just make it clear, well, you had something on this first test, we just need to do another test. It probably isn't anything serious. Whereas if the next thing is cystoscopy, or if the next thing was surgery or a biopsy, then you clearly are very worried about false positives. So yeah, I'm optimistic about the future of early detection, but the more we understand about what changes happen and in what order, and so understanding the biomarkers, what genes are being mutated, when you can detect them, and in what material, that probably is a good thing. And to just rely on a blood test for cell-free DNA or the methylation of cell-free DNA is going to solve everything. One blood test is probably a bit too optimistic.
So, Lydia, at the beginning, you mentioned that actually we're talking about 21,000, not 10,000, but that is not just bladder cancer, it's upper tract cancers as well. I noticed that Nada used bladder cancer as her specific term. And I just wondered, in these tests, are we also seeking upper tract urothelial cancers as well? So are you looking for that, Rick?
Yeah, so theoretically, the gene panel. So when we looked at the gene panel in tumor tissue, the DNA from tumor tissue, and that's the targeted deep sequencing of 1,000 bladder cancers. So the biggest sequenced bladder cancer sequencing study to date. Our urine test would theoretically identify over 97% of all bladder cancers. Bladder cancer is very heterogeneous, as Nada says. I think if you wanted to get 100%, you'd probably need to be sequencing 200 or 500 genes, and the technology to do that in urine is not there yet, but it will come. For upper tract urothelial cancer, it's very difficult to study because it's a much more unusual disease. But theoretically, based on other published literature, I think there was I think it was Cancer Cell paper last year where there was a comprehensive study of the mutational landscape of upper tract UC. If we extrapolate those data, we would theoretically be able to diagnose 98% of upper tract urothelial cancers, because there's so much there's lots of similarity in the mutations. It's just the proportion of mutations that's different between the diseases. So yeah, we're very hopeful. I think it's important to say here that in the BC Recon study, we are, Peter has had a big input. Peter Sassini has had a big input to that study, and he's, part of the successful funding application. So thank you to that. And also there will be an element of early implementation work within BC Recon from the Aberdeen group. So hopefully we've got a lot of bases covered. I suppose whilst I've got the microphone, I was going to just say one other thing. Nick James has texted and he's got a few questions in the chat. So if you can check that, thank you. And the other thing I was going to, is it for me? All right. I will in a minute. The other thing is this scanxiety question. So flexible cystoscopy, it's uncomfortable for patients. Patients probably have to take an afternoon off work if they're of working age. If they're more elderly, it might be quite a big outing. They may have to bring a relative with them. It's a big deal, it's uncomfortable, and it's expensive. But it's a relatively short procedure, 5 minutes at the most.
Reassurance.
And you get an instant result. The specialist there says whether there's recurrence or there isn't, or hopefully they do. So you have an instant result, whereas if we're replacing in the surveillance setting, if we're replacing some of those with a urine test that's posted in from home, so the patient does a sample in a special tube, posts it in, and maybe a week or 10 days later a result arrives. I don't know how you know, is that more or less, does that lead to more or less scanxiety?
Well, I'd say we'd all got used to doing COVID tests, so
Yeah.
I saw some more hands over here. So Andrew Mason, and also we've one online, I think. Yes.
Yeah.
Right.
Are you reading the one?
No, you read it. No, go on, go on.
He was talking about your urinary biomarker and was saying the key thing here is surely the NVP rather than the ability to detect cancer, as all these will need flexi anyway. A good MPV would allow patients to avoid flexi, analogous to PIRAD 1 to 3 avoiding prostate biopsy.
Yes, he's right. Yes, Nick's right. So NPV is important, and you know those are data that will come out from the BC Recon study.
So Andrew Mason.
Thanks, Rick. On your and Mika, sorry, on the BC Recon. So those false negatives that you get from cystoscopy, is that purely technical, or is there any overlap with the false negatives you get from your urine test as well? Is there anything that defines that 15% of false negatives?
I think, you know, having been in this field for a while, and, you know, Doug's probably better placed to answer than I am, you know, we kind of have a feeling that there are just some tumours that don't shed DNA, that don't shed cells such that we can detect the DNA. Now we have to go into the technicalities, our test is based around cell pellet DNA. So urine sample, it's centrifuged, cells sink to the bottom, and we extract full-length genomic DNA from those cells. Okay. Now, work that we did 5 or 6 years ago actually demonstrated that cell-free DNA in the urine supernatant, so fragmented DNA that's about 180 base pairs long and very analogous to circulating tumor DNA, that cell-free DNA probably tells you more about the tumor's makeup than those cells shed from the luminal surface. The problem is that we only get enough cell-free DNA for the assay from about 70% of urine samples, whereas we can get enough cell pellet DNA from over 95% of urine samples. And so if you rely on cell-free DNA, you're automatically, to a certain extent, limiting the ceiling of your sensitivity if you take all samples that go into the pot. And so we're looking at ways around that, and where we extract whole urine DNA. And it may be that some of those tumours that just don't shed cells and contribute to the false negatives may be tumours that are actually leaking cell-free DNA. I don't know. Doug can probably give a better answer. Prashant would like the microphone anyway, so we can give it. Doug will.
So just on that though, Rick, the cancers then that you're detecting by the urine test, you're getting a urine test positive, that then there's nothing there by cystoscopy. Things like UCAP-seq, they've seen similar kind of findings. So how much confidence would you have in the test? Like, would you say to that person to have a cystoscopy every 6 weeks, 8 weeks, 12 weeks to say, right, we're going to catch this as early as possible? Or if that isn't the case, how soon after do you start seeing it by cystoscopy?
Yeah, well, I mean, those are 2 points that need to be investigated, so I can't give you the answer to those questions. So I think for the false positives that are actually false positives, there are potentially 2 processes going on. You've got small volume disease that's not detectable. So you've got small volume disease that's not detectable by conventional cystoscopy, plus also, as you're alluding to, you know, there's the concept that normal epithelia can actually still harbor mutations, that normal tissues, usually age-related, do have mutations. So we're keen to, that's one of our next priorities, probably the next grant really, is to bottom out what's potentially going on in the surveillance setting. Because that would be another great endeavor, another great achievement if we can reduce the number of cystoscopies in the surveillance setting. I think one thing that we've sort of suggested in our forthcoming publication on the data that we showed is that perhaps the field, those who are working in this urine biomarker field, need to come to a consensus as to what is that time period where that we define a false positive as being a true positive. You know, how long after the test is exactly what you've asked. How long after the test would we expect? How long after a positive test would we expect to see the tumor? Is it 12 months, 18 months, 2 years? But I think, much in the same way of trying to harmonize, study data and outcomes, etc., we need to try and harmonize what the field considers a true false positive or a false positive. There we go. That's just completely confused everything. But yeah, so yes, how do you define a true false positive from a false positive?
So I think if you start thinking about, as a researcher, you start thinking about the biology of bladder cancer and the fact that we have very aggressive disease that maybe doesn't have a surface lesion and isn't shedding into the urine. So perhaps we have cancers that are invading very rapidly either into the bladder wall or into the vasculature. And so maybe it is a urine test is not really going to be the right way for early detection of some of these cancers. And so, maybe it's very simplistic that we think we're always going to be able to detect something in the urine that is going to tell us what's going on.
Yeah, I think you're right. Carcinoma in situ, for example, but when we look at our data, and the same with all the biomarker tests, is that they always perform better for the diagnosis of higher grade or higher stage disease. And that's what we see with our test. But yeah, you're right. Like carcinoma in situ may not be shedding cells, may not be shedding DNA into the urine, but not going the other way. Yeah, no, really great point.
There's a couple of questions.
Question to Peter and Nada and others as well. I think we perhaps need to focus what the problem is. And the reason I ask this question is, firstly, the 2-week wait or rapid access pathways were developed on a basis of a symptom. And the symptom here is hematuria. And the guidelines were based on a PPV of 3% for triggering a referral for further investigations if there is any significant underlying disease to explain the cause of hematuria. We've discussed a lot about urinary biomarkers and its sensitivity and PPVs and NPVs today. How is it actually going to apply in real life, in real practice? I'm still very skeptical because we still need to address the original problem for which the patient was referred. So let me explain this in a different way. If your biomarker test can potentially eliminate a cystoscopy, it is very good news. But will the cystoscopy be eliminated? My feeling is no, because as a clinician, there are still other reasons for hematuria. As you well explained from the Edwards paper and the other paper, at least another 10 or 15% of these patients who presented with hematuria will have either stone, UTI, enlarged prostate, XYZ. So the hematuria investigation, I beg your pardon, the flexible cystoscopy for hematuria investigation is going to stay. So I think a full review of focus needs to go in, that are we specifically looking for bladder cancer or urothelial cancers in general, or are we specifically addressing hematuria and the cystoscopy? I think we really need to focus and come up with a strategy.
Yeah.
I mean, I suppose it's about the urgency of the investigation. I should just stay up on the podium. It's the malignancies that we're looking to diagnose or exclude, isn't it? And where we envisage our test in the pathway is that it almost accompanies the referral, the urgent 2-week wait referral from primary care. So the referral goes in by whichever medium is being used, and a urine sample is sent. If the urine is positive, the patient needs to be seen urgently for imaging and other investigations. It may well be actually that their bladder is not inspected but is imaged. If the urine is negative but the patient still has symptoms, they still need to be investigated, but there's a much lower risk of them having malignant disease. So those investigations can be less urgent. So yes, we do still need to potentially look inside people's bladders, but almost there's an element of how urgent do we need to do it.
Doug?
So whilst you're waiting, I think there are two different questions. One is related to how do you manage patients with symptoms? And as Rick said, one of the issues is how urgent is it to do that? But the other is, at what point do you transfer the patient from primary care to secondary care? But the other was that there's very little progress in the pathway in terms of finding bladder cancers earlier and improving survival or reducing mortality. And so if we're going to be finding more individuals with non-visible hematuria, then we certainly don't want to be doing a whole load more cystoscopy in those, or at least I assume you don't want to be. And so I think we need to, you do need to look at that whole pathway and thinking, well, if we're increasing the amount of cystoscopy that you're going to do because you've got a screening, can you reduce it elsewhere, either in symptomatic individuals who you can get a differential diagnosis without the need for cystoscopy, or in surveillance setting, can you do away with less of it? So I think it's in general with endoscopy type things, we need to think about their use in the round and not just in screening, but if you're going to increase the demand through a screening program, can you reduce demand elsewhere? So, but I agree that, if people still have symptoms, eventually you're probably going to have to investigate it by having a look.
So I think last word over to Doug.
Okay, question for Nada. Is this a hurdle to implementation of urinary biomarkers? There are, I don't know, 6, 8, maybe 10 commercially available biomarker tests now that look at multiple markers in urinary DNA or RNA. They all have broadly similar performance characteristics, which are quite good, but it's impossible to know which one is best, because all you can do is compare lots of small unequivocal studies and sort of sum up the evidence, and the biomarker which looks best may actually be the one that had the worst studies, where they compared a 30-year-old control group with patients with muscle invasive disease. So it's not clear which one is best. Do we need to find out which one of these multiple panels of biomarkers is the best, and how would we do that?
That's quite a hard question. I think I'm not entirely sure how we would find out what is particularly the best. On basis of my understanding, I would probably say, based on its specificity and sensitivity, I would probably say that's the best one. But then if it has the worst studies, I would also have to analyze the studies and their sort of reliability. And it might be that we don't need to know the best biomarker. It might be that we have a panel of biomarkers and we just look at several. And if the patient has, those several biomarkers in the urine, they're sort of red flagged, and that would make them really urgent in the pathway. That would be maybe my understanding, but I'm not sure.
I think if maybe there were 3 or 4 competing tests that different laboratories used, but they all performed similarly, that wouldn't necessarily be a hurdle to implementation.
If they're competing, if they're a competing test, they wouldn't be a hurdle to implementation.
C or D to detect bladder cancer, and they all have very similar sensitivity and specificity. Does that muddy the waters and make it more difficult, say, for example, for the NHS to adopt a test?
The one that avoids false negatives, perhaps.
Just very final comment. What we haven't in the biomarker arena, we haven't talked about market forces, commercialization. So many of the reasons why some of these markers have not had the rigorous studies that they should have done, as Doug may be alluding to, and is that the commercial partners for the tests, or the commercial organizations that have developed the tests, they do a smallish study, they see 85% sensitivity, specificity, say that's good enough, and then they go out and market it based on one small study. We have a determination with our test to take it as far as it can go with academic-led studies, the kind that we're doing with CRUK, that then could lead into NIHR, HTA type randomized controlled trials. We have that determination to do it, and so do our commercial partners. So, we're hoping that lots of these problems that Nader has highlighted is that we can learn from them and take things forward. Clearly, that's a slower process, but you need to generate the rigorous data from well-designed, well-conducted studies, which is where Peter and Mika come in.
What proportion of bladder cancer patients would it take for a pharma company or a diagnostic company to bite? Do you think if it was like, this is really specific to 10% of muscle invasive disease, would they not care? How low would it be to get interest from them?
I don't know. I just hear accounts from others in the field who have gone on this journey and have, as soon as there's a result that looks promising, boom, the test's away being marketed. No, we don't need to do any more clinical studies. We're out. We're marketing it. We're hoping to generate robust evidence.
So I think that's a good note to finish on. We will feed Rick's passion with some food, and thank you everyone for contributing to this session. Thank you, Nader.
Thank you very much. Thank you.